Logic programs that are a result of a close collaboration between Pine Biotech, an innovative big data analytics company based in the US, and numerous academic institutions that have participated in content evaluation, curriculum review, and project design. As a result, Omics Logic programs have been thoroughly tested around the world and are known to lead to excellent outcomes for students with minimal bioinformatics background, including biology and biomedical studies majors, as well as faculty interested in bioinformatics, data science, and biomedical research. Omics Logic is an international program that is running in 5 different regions with over 10,000 users around the world. Due to this fast growth, our team is working with local and regional coordinators that are helping to refine the local program logistics and leverage our online training resources, adapting them to the needs of students and researchers around the world. The goal of the Omics Logic program is to provide training in bioinformatics, enabling independent research guided by mentors and peer example, developing a growing community with shared passion for data-driven research and appreciation of citizen science, enabling students, clinicians, and faculty of all backgrounds to develop novel and independent research using latest technology in life and data sciences. We will combine online sessions, self-guided study materials, and practical assignments for an immersive experience that has proven to be effective in our well-known and respected Omics Logic programs. Now, let me introduce you to the resources. So let us start by creating a free account on learn.omicslogic.com. So I am posting the link in the chat box so that everybody is able to come with me to learn.omicslogic.com. So if you are there, you need to click on login, and you will find that there are several options that you can get, which is you can either sign in via your email, or you can also utilize any of these social media login options that you receive. And for all participants attending today's session, we have sent you a detailed overview of how to get started with each of the platforms. You should have also the login to the TBioInfo platform. And we have also sent you links to the Omics Logic Slack account. So this is a portal that we would be using to communicate with each other. And many of our participants here are used to the forums that we have as part of the Edu platform. Now, as we are going to move to learn.omicslogic, we will be utilizing Slack for that kind of communication. And I also want to invite everybody to go ahead and check our LinkedIn channel. So let me take you all to the platform and see how it works. So this is the learn.omicslogic platform. So how many of you have been able to go to Learn? And this is free for everyone. So please feel free to join and work together with me. So how many of you are there on learn.omicslogic.com? Please put number 1 in the chat box if you are there. Great. So Aarti is there. Anybody else? Perfect. So you would notice that there is a login button. So I clicked on the login button that is right here. So everybody should get that button. So please click on login. And once you come here, you would notice this sign-in page. So if you are already a user of any of our platforms and you have a particular message, so please feel free to use the same email ID that you have been using, while others can also use any of these social media sign-in. So I will sign into the account. So once I'm signed into the account, it takes me directly to the different courses that we have. So we have a basic introductory courses that we are still launching with bytes and molecules. And as the focus for this session is genomics, we have this course called Genomics, and it is divided into several lessons. So I would really ask everyone to start with Introduction to Genomics after this session so that you can learn about what we are going to be covering. And also you can continue to explore all of these different lessons right here. And if you go back and explore the courses, you will also find that we have added lessons in Python and R as well. So everything is integrated into this one platform. And as you're going through this, you would also see that once you create an account, if you click on your email ID right here, you are able to see your achievements and your activity. So everything that you do in the platform would be tracked. Now, let me show you what we are going to be doing with Slack. So this is how the Slack channel looks like. All participants in the program have received an invitation via email to this channel. So as you see, I have posted about the first session, and Ilya has also added about the session and added an image here. So you can actually write here or ask us any questions regarding this. So please feel free to explore the Slack platform. And in case you have any questions, please feel free to ask us there. And the second platform that we have is the LinkedIn platform, which I would again welcome everybody to join. So this is the LinkedIn platform. And why we welcome you all to join this platform is because this gives you an opportunity to connect with people who are not only part of your local community, but other bioinformaticians, clinicians, as well as industry experts who belong to our community and join us from all over the world. So you can actually see the number of connections we have here, and you can extend these connections, which will be helpful for you as well. And we also have this platform on Facebook, which again, or this Facebook group that you can again connect on. So I hope that you are able to explore all of these platforms together and sign up to learn.omics Logic. For everybody else who are still not part of the Omics Logic Genomic program or any of the other Omics Logic programs, there are several free resources here on the Learn platform that you can explore. So please feel free to explore. And we are also listing here the different campuses as well as the community that we have all over the world. So you can also explore this section here. Now, let me quickly go back to the presentation and introduce you to the mentors of this session. So we have Dr. Harpreet Kaur. She's our bioinformatics trainer, and she has a PhD in bioinformatics. She will be guiding the sessions. Dr. Mohit Mazumdar is our project and partnerships mentor, and he also has a PhD in computational biology. Ilya Brodsky here is our co-founder and CEO. And now I would want to invite Ilya to talk more about genomic data analysis. Over to you, Ilya. Thank you.
Thank you very much, Pipsa, and welcome everyone to this program. I'm excited to see you all here and hope that you will have a pleasant experience today with what we've prepared. So I hope that by this time, everyone already had a chance to sign up and create your account. It is a big transition for us. We just recently crossed over 10,000 users from around the world. And unfortunately, that comes with greater demand for computational resources, which we knew was coming. And so we have been building out this site for quite some time. We hope that the new features that we're going to introduce are going to be useful and will resolve a lot of the issues that a lot of the users have been complaining about in the past. Now, to talk about this program, I want to start by reviewing how the previous portion of the program, called Bioinformatics for Infectious Disease Research and Training, really helped set the stage for what we will be covering in this part of the genomics training program. So in the first 6 months of the program, we spent considerable time looking at some introductory materials that were covering sequence variation and alignment. where we looked at multiple sequence alignment, pairwise sequence alignment, and different variations of these types of methods. We looked at evolutionary and phylogenetic analysis, where we looked at phylogenetic trees, and we looked at the relationships between viral, bacterial, and fungal genomes by analyzing alignments of full genomes or individual genes. We also looked at how to interpret genomic variation by looking at functionality and protein structure, identifying specific regions that were more conserved or less conserved, and how the interaction between proteins can help us interpret the functional properties of these mutations. And we also touched on the transcriptomic analysis of host response. And even though this program was over 1 month long, we had great sessions where we got to describe different project examples and see practically how to analyze such data. This program was actually an introduction. And so after we have completed this first introduction, we hope that it provided a perspective for the participants that joined, how they can maybe improve their projects through incorporating additional data types, by validating some of their findings using other types of omics data, and by doing some literature review on how other research projects have leveraged data sets from different repositories to combine them together and make their projects more impactful. So the transition from this introductory program to this more in-depth program on genomics is really focused on a deeper dive into the genomic data analysis. And so even though we talked about multiple sequence alignment, and we talked about phylogenetic trees, and we talked about interpreting the biological components of those mutations, there are so many things that we just did not have time to complete. And so this is phase 2 of this Bioinformatics for Infectious Disease program. It will be more focused on in-depth analysis of genomic data, and I will explain briefly what are some of the specifics that we will touch on in a few slides. But we also want to make sure that the projects that have been started and developed in the first part of the program, the project proposals, are going to be enhanced and advanced through this kind of training. So a big part of this program is not just the training itself, but continuing to work on specific project examples together with our mentors and our team to develop further the projects themselves, as well as resources, tools, and understanding around the specific areas. For example, in our last program, we talked about different data repositories and reviewed a lot of literature that has to do with the specific topics of interest that are very pressing and important in the domain of malaria and COVID-19. And we have been able to work together with our collaborators at the Tauber Bioinformatics Research Center to discuss with them how the specific project ideas could be enhanced with particular methods of analysis and particular ideas about the analysis results that we hope will make these projects more impactful. Now, for those of you that are joining us for the first time, I think it's important to understand that we have continuous training like this throughout the year. We do these projects together with our research fellows, as well as researchers, students, and faculty that come and join the research programs. And we are always interested in helping more faculty, researchers, and students develop independent projects that leverage bioinformatics and online data repositories. So to speak a little bit about the previous program and some of the outcomes, I want to invite Dr. Harpreet Kaur to share about her background and also introduce some of the things that she's observed in the previous program. So Dr. Kaur, I'll pass it on to you.
Thank you, Ilya. Just let me share my screen. So, hello everyone. My name is Harpreet Kaur, and I have a PhD in bioinformatics. Uh, Currently, I'm bioinformatics trainer with the Pine Biotech, where I'm assisting participants to learn various aspects of data analysis, machine learning, and we are also designing different research projects. So in our previous program, that is bioinformatics for infectious disease, we have covered different aspects for regarding the bioinformatic disease. So like we have also learned about understanding of the various pathogens and diseases that caused by them. And here we also covered two of the major projects, that is one is COVID-19 and another one is Ebola. Then we also learned about various analysis approach to understand their origin and finding the difference between different haplotypes. And then we have also learned how we can design our own research projects. So for that, we have learned how we can curate data from the public repository. And then we can ask a different questions like regarding their origin. Then we also learned about what could be the effect of those different types of mutations. And then we also learned about what could be how you can find the relationship between host and the uh, pathogen. So here we have also learned about different types of data like genomics data, like FASTA files. Then we also look at the transcriptomics data to understand various aspects of these infectious diseases. So here our participants have played a different, have learned lot of aspects, and they have actively shown to complete different courses, and they have also practiced various courses and as well as performed various types of analysis. So then we have one of the they have based on their previous learning, so and based on their background, so they have designed a research question. So like Kares had designed his questions based on uh, his learning from this program as well as his background knowledge. So like he has designed his research project entitled Genetic Variation Studies of Sexual Stage Specific Antigen PFS 230 and PFS 48/45 of Plasmodium falciparum population in Africa and their potential implication on transmission blocking vaccines. Apart from Curry's, there are various other participants which have designed their research questions based on their learning as well as their background. So they have tried to address different research questions. So we are looking forward how you can actually, even in the upcoming program, how you are going to implement and complete your research projects based on the genomic data analysis. So I'm looking forward being your mentor. So most of these projects are actually have we have reviewed these projects, and we have asked few of the set a few of the criterias. Based on first criteria that we have observed what kind of question that they are going to address, then whether this question is actually novel enough for a public, and then whether the data set that they are analyzing that is good enough to address that question. And next our criteria is to find out what are their analysis approach, so whether they have the sufficient knowledge about the all the algorithm uh, which they are going to use to address their question. So eventually we have find 4 or 5 very good projects depending upon our the criteria. So we are looking forward in our upcoming program that you will able to complete these research projects and also learn a new aspect like where you can use a genomic data to address your question. So now I would like pass this session back to Ilya. So over to you, Ilya.
Thank you very much, Dr. Harpreet. And hopefully this also helps us understand how this program is going to help you further advance your project ideas and continue your research. So, but before we jump into how this program is going to be structured and what are the different topics that we will cover, I want to pose a question that might be relevant to all of us interested and maybe involved in research related to infectious diseases. So in 2017, what has been shown through different statistics gathered from around the world is that communicable maternal, neonatal, and nutritional diseases, all kind of bundled together, but they include infectious diseases, have been steadily on the decline. And you can see that the primary focus was actually on non-communicable diseases, such as cancer, diabetes, and others. And what's interesting about that is that the health systems around the world, including a lot of research funding, has actually been diverted from infectious diseases to the challenges associated with these other types of diseases, like chronic diseases like cancer and others. And what has changed, obviously, in the past year is that all of a sudden, we see this major rise in mortality and prevalence of an infectious disease that shows how these various comorbidities, including heart disease, cancer, and other types of respiratory diseases, for example, are all major factors to consider, but also in the context of infectious diseases. And so this, I think, is an opportunity for all of us to think again about the centrality of infectious diseases, the importance moving forward. I think we will all see major changes in funding mechanisms, specifically designed to address emerging infectious diseases around the world, including in Africa and Asia. We will see a major focus on genomic data and NGS data in general that has become so central to this COVID-19 outbreak. And we will see how this will provide an important opportunity for those neglected tropical diseases as we see their centrality to how susceptible different populations are to COVID-19 and other infections, and how they change our understanding at the basic biological level of what are the mechanisms and processes that we should consider for prevention, treatment, and intervention for different types of emerging infectious diseases. And so, in fact, if we look over the history, I think what's very interesting is that these outbreaks, endemic and pandemic outbreaks around the world of infectious diseases have caused these major loss in life throughout the centuries. And in the past century alone, we see in the past maybe 50 years or less, we see these major outbreaks happen again and again. So you can see here, just in the last part of the slide, right here, starting with maybe Spanish flu in 1918. We see then Asian flu, Hong Kong flu, then we see HIV. We see SARS, swine flu, MERS, Ebola, and finally COVID-19. So obviously these numbers here are a little bit outdated, but it really shows how in the last century, there has been an acceleration of infectious disease outbreaks caused by pathogens that might be even not considered alive, right? Because viruses in many cases cannot be categorized exactly as living. And so to learn about the different measures of intervention and the way that these diseases have been addressed and prevented and treated in the past, we can look at the history of different measures that were developed. For example, how specific measures on the public health side, such as first continuous municipal use of chlorine in water in the United States, or how we can see specific novel treatments like penicillin, and how we see specific vaccines being designed over time, how these interventions that were really brought forth by the scientific community have helped us establish an approach to diagnose, identify specifically the cause of a disease, to understand the basic mechanisms of disease, and then to look in nature for existing interventions and responses that could be enhanced or repurposed for a particular challenge, have been used in the past 100 years to really limit the number of deaths. As you can see right here, the rate of mortality from infectious diseases. And so to look at these specifically in detail, we're going to continue looking at several examples that include Ebola, different types of infectious diseases that emerge as a result of biotechnological advances, how we can look at data coming from the novel coronavirus and associated coronaviruses. We will also take a look at bacterial infections like tuberculosis, where we see an emergence of multi-drug resistant strains of tuberculosis. So we will actually take a look at a lot of the applications of genomic data analysis in the context of these projects that were sourced from publications that play a major role in infectious disease research around the world. So here are some examples of projects that we already started talking about and we will continue to review using additional methods of analysis and learning about how data analysis approaches can help us explore, understand, ask specific research questions, and make sound conclusions that could be presented in the context of a research challenge that we're trying to address. So one of them that we've kind of started developing in the context of this first part of the program is how does bioinformatic analysis play a major role in understanding of malaria, the diversity of the pathogen, the interaction between vectors, pathogens, and the hosts, and how we can use that understanding to really evaluate the efficacy of vaccines that are appropriate at different stages of infection, how we can measure the interaction between different proteins and understand how maybe that interaction could be intervened in, as well as to understand the different vaccines that are designed to prevent transmission, to also prevent infection, and what can be done in the context of the low rates of efficacy of vaccines that have been seen up until now. We will also speak about the emergence of selective pressure outcomes as a result of the introduction of novel vaccines and antiviral treatments for SARS-CoV-2. So we already know that the vaccines are being rolled out around the world. We know that there are asymptomatic patients and patients with a suppressed immune system. We also know about the variability of infection around the world. And some of the questions that we're going to continue to ask is how do these selective pressures change the virus itself and the population dynamic that we are so familiar with seeing as the pandemic continues. So we'll try to think about what is the future of how these different clades that have emerged over time and in fact have diversified this pandemic from just a single strand to multiple strains of the virus, and how the interaction with the host immune response, the different treatments and interventions that are being introduced will change the course of this pandemic, and how we will start looking at zoonotic transmission over the next decade to address these emerging pathogens. One of these pathogens that I of course mentioned, Ebola, has seen a continuous series of outbreaks throughout the past couple of decades that really shows that one of the pressing challenges is the changing ecological context of this disease. So in fact, the prevalence of Ebola in a particular region and the interaction between different animal hosts that have been contained up until now and maybe not as exposed to interaction with humans, as well as the changing climate, might give us some sense of what are the factors to consider as these pandemics are bound to happen once again. And we'll also take a look at a very fascinating history of the vaccine design and antiviral drug design for these novel pathogens that have been produced in a record time. So we see this with COVID, but a big part of what we see with COVID is actually the result of what started happening with Ebola that happened just recently in 2014, 2016. So we're talking about less than 10 years ago, where Ebola outbreak was actually one of the major events that that gave a central role to next generation sequencing and genomic data analysis. And in fact, we see how genomic data that is cheaper to produce, easier to analyze, and people are becoming better and better at making sound predictions about the importance of the different changes will actually accelerate the field of bioinformatics in application to genomic data, specifically in the context of vaccine design, drug design, and prevention. And so another important area that I already mentioned is the emergence of multi-drug resistance with tuberculosis. And multi-drug resistant tuberculosis is actually prevalent in very specific regions that include Russia, Asia, like China, Pakistan, India, Bangladesh, South Africa, Nigeria, and other African countries, as well as in South America. And so what's interesting is that we have a lot of information about the way that these tuberculosis strains have been treated. And so understanding the basic mechanisms of resistance, as well as the interaction with those drugs, can help us study genomic data and find important associations between genomic variation and what we know about drug susceptibility and resistance of Mycobacterium tuberculosis. So we will actually take a look at specific types of antibiotic function, how they interact with the different strains, the emerging mutations in those strains. We'll take real data sets and analyze them, and then perform genome-wide association studies to find associated variants with that drug resistance. And we'll also think about how similar approaches could be applied to SARS-CoV-2, could be applied to malaria, could be applied to other diseases as well. So to kind of summarize, what we will do in this program is we will spend another month learning about different approaches to bioinformatics. These approaches are going to be used by the participants to enhance or develop further their bioinformatics research studies and work with our mentors to really see how these projects could be, maybe in collaboration with others or maybe independently, could be completed at the next phase to maybe make those projects again into presentations to our peer community to get some feedback and expert advice, and potentially complete some of the exploratory and comparative analysis that we will do using some of the tools that we will learn about. So what are these tools that we're going to be using? Well, as you probably can imagine, there has been a significant growth in the number of data sets that are available for anyone to be able to analyze. So what you can see here are a number of different types of sequencing technologies, Illumina MiSeq, HiSeq, and others, including sequencing with Oxford Nanopore and with PAC Pacific Biosciences, PacBio. And so we see both an increase in the number of data sets that are available, but also in the types of data sets that are available. Targeted sequencing, whole genome sequencing, amplicon sequencing, and many others. And so what we'll try to do is we will introduce you to additional methods of analysis on our big data tBioInfo platform, where you will be able to analyze hundreds of samples with large quantities of data, performing in-depth and complex analysis with a focus on integration, because ultimately what this platform provides is an ability to process, analyze, visualize, and integrate multiple types of omics data. So what are the types of data that we're going to be speaking about? First of all, we will continue to speak about sequence alignment, both in the context of FASTA files and FASTQ reads, because there's a lot that could be done as we start to understand the differences between different methods and the specific outputs that are important for the different methods of analysis. One of the things that we will look at are the specific types of analyses that allow us to understand relationships between sequences and study the structure of identified relationships in the context of a population or in the context of a comparison between multiple types of phylogenetic relationships. So we'll try to take different types of phylogenetic trees and analyze them for how well they represent what we observe in data. How do they explain the relationships between different strains that are related or not related? How they could be applied to different geolocation studies so that we can analyze populations and associate them with phenotype. We'll also take a look at a detailed analysis of raw FASTQ data sets that contain millions of reads and show a lot of variation that is lost when we just look at consensus sequences. So we'll take a look at such important information as mutability, or how frequently we observe different mutations in different types of populations. We'll look at coverage between different regions and how this important metric can help us make comparisons and understand our data in the context of the limitations of sequencing and the opportunities for high coverage, consistent quality regions that we observe in our data. We'll also look at the application of such information to the study of interaction with the immune system and how we can use conservation, epistasis, mutability, and other types of factors to categorize different regions and their susceptibility to genomic variation. Some important tools that we will discuss are the analysis of mutations visually so that we could explore specific regions that we have understood, how to compare different regions of mutations between groups of samples or individual samples, like in germline somatic mutations in cancer, or like mutability and variation from the reference genome and the specific haplotypes that we observe in infectious diseases. We will also look at other types of method of genomic analysis that will allow us to ask specific questions for the host response and also for the interaction between the pathogen and the host. And also we will look at how diversity of some populations of viruses, bacteria, and pathogens can help us interpret FASTQ files that have been produced as a result of sequencing pooled data that is often happening when we take samples from individual patients or pool data from multiple patients and they're not demultiplexed. We'll also talk about genome-wide association studies that are important for us to understand the impact of multiple mutations spread across different regions of the genome as they work together to produce an observed interaction with treatment or with other types of important phenotypes. Now, what's important about this data, as Harpreet has already mentioned, is that a lot of this data has been made available. And what's important to understand is how to properly navigate different resources, understand the specific types of data sets that are available, look at the quality and the metadata, and organize our data to structure it in such a way that we can answer specific research questions. And so we have already looked at a variety of malaria repositories like MalariaGen and PlasmoDB. We've looked at NCBI virus, where we can find information about viral strains and genomic variants associated with specific types of pathogens. We will also look at different repositories for bacterial infections. And so what we will try to do is schedule a way to prepare our data set, organize it, and describe the metadata, and to be able to think about what types of outputs from analysis are expected so that we know whether we can answer a specific research question. We will also spend some considerable time working with protein structures. And there we will see both the specific kinds of properties like hydrophobic or polarity or electrostatic, and think about the interaction between different portions of proteins, complexes, and proteins to other proteins, as well as do docking and interaction with specific ligands, where we can study the specific mutations and changes in amino acid properties and the interaction with molecules and other proteins. We will also look at the response from the specific treatments and think about drug repurposing so that we can analyze data that can have an important component of genomic data analysis as we try to interpret how the drug response and the treatment response and outcomes could be mapped to genomic and transcriptomic data. So to summarize, we will look at viral genomics and evolution, revisit phylogenetic trees, multiple sequence alignment, and analysis of raw FASTQ reads. We will look at the epitopes so that we can understand immune responses and how genomic data can provide us with insights about the interaction with the immune system. We will think about the community of viruses and how they work together so that they can use the diversity of that population to overcome immune responses. And we will also think about pathway analysis to measure and interpret biological significance of found variants and the interaction between proteins that we have identified. So let me now share a little bit about the schedule of this program. Again, the program is called Genomic Data Analysis with a focus on infectious diseases. Beepse, if you could share once again the link to the registration page. So for those of you that are joining for the first time today, we have this link called genomics.omicslogic.com. There you will see the schedule, and I will go through the schedule to explain what are the specific sessions and what will be covered in those sessions. But before I do, I want to ask whether you have any questions about what I have asked so far, or what I have spoken about so far. So are there any questions before we move on? Okay, thank you, Mohammed. Okay, so if no one has any other questions, I have my first question to you. So for those of you that are in today's session, how many of you actually have already created an account on learn.omicslogic.com? If you have, please put number 1. And okay, thank you, Ayurinde. Again, for those of you that have joined from a previous program, your same account information from Omics Logic should be available, should work. Okay, excellent, thank you. Okay, and I want to also ask, okay, so my second question is, and instead of putting number 1 in the chat box, if you could put number 2 in the chat box, if you confirm, or no if you haven't, right? So if you have already started working on a research project, please put number 2, okay? So if you have started working on your research project, okay? And if you have not, put no. Okay, thank you, Courage. Okay, Brenda, okay. Okay, excellent. So for those of you that have not yet started on a research project, this is going to be the opportunity to learn from your peers that have already started their research project. So Courage, Abukari Zakaria, Mohammed. So some of you are already working on some research projects. For you, this is our opportunity to meet new people that might be able to contribute to your project, enhance it, and maybe introduce new questions that might be important to ask. For those of you that have not started a project, this is an opportunity to learn from those that have spent the past month or so developing a research project and identifying a data set, refining that research question, and proposing a specific project that has specific competitive characteristics that will continue in this session. Okay, so Beepsafe, whenever you're ready with the poll, let me know, and then in the meantime, I'll go through the schedule. Okay, so first of all, we will start reviewing some important information about mutations in general, but also how they play a specific role in infectious diseases. And so in infectious diseases, as you might imagine, they play a role in two ways. The first one is genomic variation of the host. And the genomic variation of the host is an important part of our understanding about variation in population response to disease. So for example, we know where there's a prevalence of malaria infection, sickle cell disease, or in different types of populations with diabetes and their change in weight and other types of kind of predisposition or comorbidities can have a very important difference in response to infection. So we will take a look at genomic variation, kind of clarify the terminology, make sure that this terminology is clear from project examples where we will see how this terminology will help us understand and study important questions. So then today, we will actually introduce the different resources, and hopefully most of you that are joining the program have already completed your registration. But we will cover how the specific elements of the program fit together to advance your research project. And so we'll talk about the project examples that have been prepared as case studies for you to learn from. We'll talk about pipelines, and today at the end of this session, we'll take a look at one of these pipelines that we want to run. We'll look at the coding component where you can learn to run and analyze a data set and visualize it, or maybe look at a statistical test in R or Python. And we'll take a look at how the sessions that we will cover in the next couple of minutes will lead to a greater appreciation for what the genomic data can provide in the context of your research question. So one important thing that we'll focus on is how the emerging centrality of next generation sequencing is actually changing infectious disease in general and medicine and clinical applications of analysis of genomic data. So like most of you know, a big part of working with such data is being able to code and automate some of your analysis. And so to illustrate this, and for those of you that are already familiar with this, I want to show you one of the courses that we will use as an example to look at genomic variation. So to do that, let's go to learn-omicslogic.com. So please let me know once you're logged in. I'm just going to log in here real quick. Okay, so as I mentioned, you can use different types of accounts here. You don't have to create an account, or you can register. So let's go in. I will use my Google account to register and log in. And what you'll find here is that we've tried to rearrange some of these resources so that you can easily access different things in the order that we have kind of prepared for this program. So you'll notice here that we have some courses that are introductory, and then some courses that are project-based, and then we also some code-based analyses here. So for example, Python coding and coding with R, getting started with bioinformatics and things like that. So what we'll take a look at is this course on genomics. Now, in the course of this program, you're going to see some new materials added here about different statistical tests, machine learning approaches, and examples of different projects that we're going to be covering in this session. So you'll also see how different courses or lessons inside courses have this tag for learn, which means that it is using the platform with a user-friendly interface and introduces some theoretical material. Coding in R will provide you with some exercises that you can complete and see that you've done the assignment correctly. And then you'll also see a coding with Python. So how many of you are here on this course? I'm just going to paste the link here. And I want to demonstrate one of these, just how it works. I know that some of you are already familiar, but please let me know once you're in here. So again, I pasted the link in the chat box. And this is a free course, by the way. So you'll see that the lessons that I will show you today are completely free, so anyone can do those. So please put number 1 when you're in. Okay, thank you, Courage. And we will go into this lesson called Working with DNA Sequences. All right, excellent. So in the context of R, it's an object-oriented language, and we can deal with sequences as objects, asking questions about the comparison between them and trying to identify specific differences when we know that the context is that overall these sequences are comparable or very similar to each other. So here you can see that one of the first things that we'll review is pairwise alignment. And one of the important kind of parts of this learning is that you don't only learn about the problem, but you also get to try it. Now, some of these are as simple as just pressing the run button, and it will run in the background and produce this output. So you can see global pairwise alignments, single subject, 1 of 1, and then you can see the pattern, the subject, and a score. And this is a similarity score, and you saw the little fireworks come out at me at the screen, and that means that I've done this correctly. And so you can see here that this introduces us to some basic ideas about how to work with R. We have to call a library. The library contains specific functions. We have to provide it with a data set that is stored as an object. So we have here 2 objects, DNA-seq1 and DNA-seq2. And then when we perform multiple alignment, we also print it at the end. And that allows us to test whether we have done it correctly, but also to see a score of alignment. Now, this is the output. And now we can try another a different comparison of sequences, right? So these sequences were at a score of 102.68 something. And now we can run another one, which is going to be a little bit different. Okay, so again, I get the fireworks, but you can see that the score of my analysis is much lower, 55. And so a natural question would be to say, okay, well, can I look at the specific sequence, sequence 1 and sequence 2, and try to understand why is this difference? And so the reason is that obviously these are different lengths, okay? And so there's different lengths here, there are different lengths there, and I can start looking for, okay, well, what are the specific changes maybe in the beginning, right? You can see ATGG, ATTTT, right? So this has much better similarity overall, even though the lengths are also different here. So as you go through these different types of assignments, eventually you will be introduced to a project. And so, for example, here you get a small assignment. These assignments, again, you can run them to understand how they work. Okay, here you see some basic alignment. Here you will see some kind of a visual visual result that you can check. Okay, and instead of looking at the console output, you'll actually validate that there are specific changes, right? And we can forget about everything else, and we can just look at the specific changes in the sequence, and so forth. So going through these assignments, you will also get introduced to how this is relevant to a biological problem. And the biological problem that we are looking at here is metabolism of a specific drug. Now, metabolism of drug is an important component in what's called pharmacogenomics, right? So when a pharmacist gives a drug to a patient, they want to know that this drug is going to be metabolized well. And so you can see how specific mutations, and now we need to think about what types of mutations are out there, have an impact on this basic pharmacological aspect of assigning treatment to a patient. So these concepts are built to combine the specific practical skills like R with an understanding of genomic data and how it relates to biological understanding of DNA variation, and then gives you a glimpse of the importance of such genomic variation in a variety of applications. So I just wanted to make sure that it's clear what that really entails. Okay, so these are going to be some examples of our scripting and working with specific libraries and methods of analysis of small data. Now, we will also look at how the specific variation in larger data sets, and here you can see a number of whole genome sequences in FASTQ files, could be analyzed using a visual report. Okay, so this is actually also a practical assignment. It's considered to be basic because really you're working at consensus sequences and the result of a comparison of multiple longer sequences that has been prepared on the T-Bioinfo platform. So to talk about that, in our next session, we'll talk about mapping reads, addressing technical variation, and performing what's called per nucleotide analysis. So when we talk about per nucleotide analysis, what that means is that we take individual reads from the raw FASTQ files that are often stored as sequence read archives, and we want to understand what are the specific changes. As you can see here, not all of them are going to have a huge impact on the consensus, and not all of them have an equal influence on what we consider to be a SNP, a variant, or a mutation that causes a change in the amino acid sequence. So we'll really think about how FASTQ files and the raw reads could be used to analyze clusters of changes in a number of reads, combinations of reads that represent haplotypes within a given sample, as well as the consensus sequence that might change as we try different types of reference genomes and as we address the technical variation by trimming reads and also eliminating mapped regions onto the host genome. That's also an important part of the type of variation that we will find. We'll also talk a lot about mutation analysis. So oftentimes the problem statement sounds like this. Here's a set of mutations that we know contributes to drug resistance, for example, in the context of malaria, to chloroquine. So if we know those mutations, how can we actually look for those mutations and characterize the nucleotide sequence variation that we observe to what's called variant calling? Now, variant calling has several different specific parameters to it. When we look at calling variants that are present in all of the reads, when we look at variant calling present in a portion of the reads, or when we look for specific variants that we know how to interpret and forget about the rest. And so we'll talk about differential mutations, how those variants could be called and analyzed together with what we know from a reference or an annotation database, and also how do we interpret the biological meaning of some of those mutations in the context of other mutations that are present throughout the genome. In our next session, we'll talk about allele frequency, and we'll talk about variation frequency and the continuous data that allows us to study large data sets without looking at the specifics before we've identified what is appropriate and significant for a data set that we're studying. A good example for this is this example from the Ebola virus, where we took the per nucleotide analysis. We created mutability scores for each position, for each sample and position. We grouped our samples into specific geolocation-based groups. And now we're asking the questions, how the specific mutations in a gene or the whole genome can characterize subpopulations of these viruses or parasites. This is an example from malaria that will have a significant effect on the variation, right? Because we're looking at the characterization of the population based on mutations, individual positions, abundances of mutations in particular regions, or just the conservation of particular regions. Maybe that has an importance for their specificity to the population. We'll also talk in depth about differential mutations that have that we need to understand in terms of the statistical significance of the parameters that we have in a given data set. So for example, we already talked about coverage, we talked about mutation frequency, and now we're going to combine the two to perform some advanced statistical analysis of mutations that are either more prevalent in a particular a group of samples or in another one, right? So it's kind of like differential gene expression, but in application to mutations. And so we'll take a look at some of these examples, that we will apply them to genomic data from Ebola viruses, from malaria, and from other pathogens. We'll spend also some additional time speaking about the sequence and structure relationship. And so what's important and what we have not really covered in the previous part of the program is how, with the growing number of data sets that are available, what is needed is the integration of these different parameters to quickly go through genomic variation, relationship at the phylogenetic level, association with different phenotypes, and the structural components that allow us to ask questions and make interpretation quickly. And so what we're hoping to achieve during this session, by the way, this session is going to be available for anyone that would like to join, is to prepare a collection of impactful examples using malaria, COVID-19, polio, and other types of data sets that we have been working with over the past several months, so that we can provide an important view of data linkage and examples where we can rapidly explore data and generate hypotheses based on the available data quickly. And so hopefully we will create a resource by that time, together with you and your collaboration, with annotated samples, with important phenotype-genotype relationships based on phylogenetic analysis and mutation analysis that could be explored by others that join that session. An important part here is to be able to look at various statistical properties like correlation, frequencies, analysis of variance, and perplexity that will allow us to capture important regions that are more or less susceptible to the different types of genomic variation, to be able to ask questions about phenotype, right? Because oftentimes our hypothesis is driven by knowledge and not by exploratory data analysis. And so we will focus on some of the methods of exploratory analysis, like principal component analysis, like statistics for mapping and normalization, as well as analysis of regions that are more or less susceptible, so that we can gain a snapshot of big data sets that we have to deal with that contain thousands of samples, and we have to make a selection about specific variation associated with conditions. In our next session, we'll talk about biological interpretation of population structures, immune pressures, and rates of transmission as we look at some of the characteristics found in visual representation of genomic data, such as phylogenetic trees, relationships based on clustering, or analysis of multiple sequence alignment scores. So we'll look at how we can use this numeric data to inform our biological interpretation and understanding of data. And as I mentioned, we will also spend some additional time on analysis of 3D protein structures, looking at important proteins responsible for the process of infection, like the SARS-CoV-2 spike protein, looking at interaction of different proteins responsible for different sexual stages of malaria pathogen, looking at different types of changes that the proteins undergo inside the cell in the process of infection. And then we'll also look at genome-wide association studies, where we will look at genome-wide association methods and what the models of genome-wide association can tell us about multiple positions across the genome associated with a phenotype, as well as thinking about such topics like drug resistance and evasion from the immune response. And finally, we will have a free webinar that is going to be available again for everyone to join, that will be focused on structural computational biology. And we are hoping that by that time, many of you will give us enough kind of examples with your research projects that we can incorporate those into a review session with some application of molecular dynamics, docking similarities, and look at the relationship between genomic data and protein data that has an interesting application and context, and also has been placed at the right place in the project, in the research projects overall. So this is just to give you a glimpse of what will be covered in the program. I know that we're already running over time, and I'm sorry that it's taking a little bit longer, but we do have the poll ready. And now I would like to ask that you also help us understand a little bit more. Beepsa, if you could launch the poll. If you could help us understand a little bit more about your background, so which country you are from, about your educational background, as well as why you are interested in learning genomics. You know, if you work in this field, or you know, if you're just interested, or if you're just new and want to learn more, as well as how you would rate this session. So again, this is an introductory session that we prepared so that you will be familiar with the different resources and the topics that we will be covering in the upcoming full program. And if you have not yet participated in other webinars, whether you would be interested so that we can appropriately send you the details for the upcoming sessions, some of the sessions that we mentioned today, some of the free webinars that are coming up, as well as some of the review sessions of research projects for those that will complete this program in the next month or so. So again, thank you for giving me this opportunity to provide you with this review of the program. And I will pass it back on to Beepsa to conclude with some details about registration for those of you that have not finished your registration process.
Thank you, Ilya. So I have closed down the poll right now. So for everybody who has not yet registered, let me show you how to register for the program. So this is the registration link again. I will post it one more time in the chat box for everybody else who has not gotten the link. So this is the link again to join the program. And once you're on this page, as Ilya mentioned, you have an introduction to the program. You can watch this video that we have prepared to introduce you again to what this program would be covering. And if you scroll down, you would be able to see the different sessions that we have. So all of our sessions on each of these dates are timed at the same time, which is 9 a.m. CST. And you will be able to see even the free webinars which are listed here for the different sessions. So please feel free to go through this list of the sessions that we have here. And so how do you register for the program? So if you click on Get Started, you would be coming up with 3 different options to register for the program. And again, to clarify for everybody, the beginner option gives you access to all of the resources that are that we spoke about today and that are available to all participants participating in this program, while the intermediate access gives you the chance to work one-on-one with a mentor and work on your project or any other area or questions or doubts that you may have. So if you find that you want to have a one-on-one session or are interested, you can join this. While the advanced license takes you a step further, so if you would want to go ahead and explore more, which gives you the opportunity to work on your own research projects with an additional 30 days. So the advanced license gives you an additional 30 days to work on your own project with our group here, who is already working on the several different projects that Dr. Harpreet showed earlier and Ilya has also spoken about, and also the research fellows who are part of our program. So please make sure that you go through these three options, and whatever is suitable for you, you can take that option and apply for the program. All of the basic resources that we discussed, like the Learn platform, the Slack forum, as well as all of the other resources like the Server TBA Info platform, would be available to everybody who is joining this program. The only difference is the additional resources that you can utilize or the additional access that you can get. And once you choose either of these options, you can click the select option, and here's the payment details that you can enter, and you would be registered for the program. And please make sure that if you are somebody who would want to request for a scholarship, we also have a scholarship option. So I would just send you this link again. Let me post this link in the chat box. So this is only available for a very few people. And if you want to join this program immediately, the next session, as you saw, is on March 16th. So you still have some time. All you need to do is fill up the form that you have at the end of this page. Please make sure that you're mentioning your references, and we would be setting up a meeting with you and one of your references to discuss the opportunity. And based upon that meeting, you would be notified whether your scholarship has been accepted or not. So if you are a student or anybody else who would require a scholarship, please feel free to set up this meeting and fill out the scholarship form. And I would again like to invite all of you to go ahead and join our, join our groups. So let me go back to the groups one more time. And in these groups, we try to be as interactive as we can. So please make sure that you are also reaching out to us as part of these groups when you are joining them. So thank you so much for sharing. And also this group in Facebook. And for all participants, again, I want to invite you all to join Slack. So you would have received the Slack invitation. It is important that you accept that invitation and start posting any questions that you may have here on the group. So this goes for everybody who is joining the program. So once you're registered for the program, we will send you an invitation to join this Slack group using the email ID that you have shared. So please make sure that you are there in Slack. You are able to use Slack. In case you're having any issues, please let me know. And if you are someone who has just joined the platform, please save my email ID, which is marketing@pine.bio, as I will be sending you regular updates regarding the sessions as well as the programs. You would not want to miss those updates. So please make sure that you have the email ID saved up and you can see this. Also, we have support@pine.bio, which is the email ID of Dr. Harpreet Kaur, and you can reach out to her regarding any of the technical questions that you would have once you start going through the courses and the resources here. So please feel free to save both of these email IDs for future references. And thank you all again for joining today's session. This session is also available on YouTube, and we would be emailing every participant regarding this session again. And I hope to see you all in the upcoming session next week. Have a wonderful weekend, everyone.
Thank you, and looking forward to interacting with you soon as well.